For coming today. I think we have a wonderful talk on a fascinating subject to listen to. And I'm really delighted to have Calum MacRae as our speaker today. Calum is a professor of medicine at Harvard and does both research and clinical work at the Brigham. I never know how people do both. One thing that I'm sure you all know from the title of his talk is that he likes fish, and in particular zebrafish, because they're transparent and you can see the heart, and image the heart and the embryo during evolution. And this has been something that Calum brought to our field of arrhythmias and development of the cardiovascular system in the heart specifically. Calum is also the head of genomic medicine. Is that right?
So it's a clinic that focuses on the whole genome and the whole exome. So that's basically for diagnosis.
All right. Well, it's all yours.
Well, first of all, I'd like to thank Professor Rudy for the invitation to come here and for really a fantastic day. It's an honor and a pleasure to visit WashU. I've really had a fantastic entire day, and it's been great to speak to everybody. Thank you for everybody putting up with me and my silly questions. I need to, for some reason my disclosure slide, I see what happened, I left it in here, but I do have patents in genetic testing of cardiomyopathy and cardiotoxicity testing in fish, and obviously the biggest conflict of interest is academic self-interest. So I think that's actually one of the most important things for me to know. We really came to the zebrafish about a decade ago, largely because of a block in the efficiency of there we go. Is that better? In the efficiency of in vivo modeling. We recognized that there were these enormous tracts of genomic information, functional genomics. There was a whole host of cellular data on differentiation and regeneration. Even the things that we thought were simple were incredibly complex. Every channel was really this massive macromolecular aggregate of proteins and lipids. People were building these incredibly complicated networks, and yet we were still modeling relatively slowly, one gene at a time. And we needed to think about ways in which we could change the scale of investigation from this sort of relatively simple deterministic approach, which is how I started working in human genetics, to a much more generic systems level analysis where we could study potentially all of the genes at the same time, all of the other non-omic elements at the same time, and look at them under basal and perturbed states. And the perturbations could be any of the things that you or I might imagine: genetic modifiers, environmental modifiers, diseases themselves, which may change the way in which everything works, drugs. And importantly, integrating across molecules, organelles, cells, organs, and then up into the whole animal and even into populations, and trying to develop some generic solutions for getting at the underlying biology. As I pointed out, actually this slide doesn't project very well, but a couple of figures are missing, but in essence, the key element is in the middle here, which is there are also a whole host of things that are never measured in any of these global experiments. Calcium, obviously, is the most obvious one. Even since the middle of the 18th century, calcium was recognized to be an almost ubiquitous modulator of most biological processes. But you can see here a whole host of other, I've only obviously scratched the surface of this list, variables that are just not measured in most global experiments. So we were thinking about what the most useful model organism might be to tackle this. And we had, not alone obviously, but we realized that the zebrafish, which is seen here, and which this is the first 18 hours of development, so you can see it fairly quickly go from a single cell to a recognizable vertebrate body plan, was actually a useful tool. It had been used to some extent, in fact quite extensively, in developmental cell biology, largely because of its transparency and its relatively efficient developmental timing. But it had some very important characteristics that I thought were useful for building this type of global analysis of physiology. And that those were, it has a diploid genome, which is actually it's one of the few transparent fish that does. It's outbred, and so it would actually be representative of much of the human genetics that we're used to in encountering. As a result of extensive efforts of the NIH and the Wellcome Trust, genetic and genomic resources were accumulating fairly rapidly. And then, in addition to the genomic series that were being identified, there were also large chemical screens beginning to be done for developmental purposes that could be accommodated for other biological purposes in large scale. And importantly, all of this can be done in 384 well plates. So there are actually 4 fish. You can really 4 or 5 can survive in a single well of a 384 well plate for up to 7 days, certainly 5 days. So you can begin to imagine scaling in vivo biology, in vivo genetics, in vivo pharmacology, integrated physiology, cell biology, anything that you can phenotype at this scale, you can begin to imagine screens. And in fact, I'm going to show you, I hope, that a lot of this single cell biology, fundamental cell behaviors, some of the fundamental elements of organogenesis and chemical biology are all accessible in single wells. We can manipulate genes within a week of identifying the gene that we're interested in targeting with morpholino oligonucleotides for knockdown. Transgenesis can be done even as transients, particularly for the heart. Most of the things that we want to do in the heart can be done in transient transgenesis. So it really accelerates the timing and changes the scale of what's feasible. I'm not going to talk about most of these today, but in fact I'm going to focus the whole of the rest of my talk on arrhythmias because of the audience. And in fact we deliberately picked arrhythmias because of the unique features, being able to go from single dipole moment in the membrane all the way up to integrated physiology and even population biology with the same tools. I think that's one of the great advantages for integration that and modeling that arrhythmias and electrophysiologists really have access to. But we have done a whole host of other phenotypes, which are seen here: vascular biology, some tissue-specific molecular imaging. These are enzymes in the intersegmental clefts. We've done behavior with drugs. We can do a whole host of contractile physiology. We've even done just random phenoclustering, where we've built a panel of 130 phenotypes that we automatically can accrue in the first 72 hours of development, so that we can begin to scale almost everything that we do to the point where it's accessible at this scale. Metabolomics, we've done a little bit in conjunction with a group at NGH run by Rob Gerson, and then simple things like this is calcification in the otoliths and in the vertebral bodies of the fish. So all of that is accessible. The reason that we wanted to do this really is, and I alluded to this a second ago, is to be able to do direct screening, to use in much the same way as people have done with antibiotic selection for bacteria, to do that for complex vertebrate phenotypes, for repolarization, for automaticity, for conduction defects. Can we build those types of phenotype-driven screens so that we know we're pulling out the genes that are important for this, the small molecules, the environmental factors that are important for this in a directed and essentially unbiased fashion, and hopefully do it in a saturated manner. So we've built almost everything I'm going to show you this afternoon is built usually in 96 or 384 well plates, most commonly in 96 because of some of the resolution. We have a completely automated screening system, and most of the phenotypes I'm going to show you today, in fact all of the subsequent ones, are based on the fish heart, which I should note is a 2-chamber heart, so atrium and ventricle. And yet I hope to convince you that there is actually a remarkable amount of homology, particularly at the level of membrane behaviors, but to be honest, across the entire spectrum between this and the human heart. When we first started doing this, one of the first things that we asked ourselves is how can we begin to A, validate the biology of the fish, and B, show that it's worth actually having a whole organism to do this. So we picked a phenotype that we knew there had been issues with single cell approaches. There had been a necessity to move into whole animals, and where the complexity of the underlying biology was understood at a molecular and at a cellular level. And so arrhythmias were an obvious place to do this. I am actually a cardiologist, but although I've done actually quite a lot of electrophysiology in the renal system a few decades earlier, had only begun to work in cardiac electrophysiology about a decade ago. We liked repolarization for two reasons. It was a big regulatory problem with the FDA. There's clearly, as everybody in this room I'm sure knows, remarkable complexity emerging in terms of the local organization of ion channels and their partner proteins, as well as their partner lipids and other small molecules. In addition to this, it was already recognized that there's tremendous heterogeneity that's responsible for many of the arrhythmia phenotypes that are important clinically, and particularly in the case of toxicity, there were known gene-environment interactions, and thus an implicit need for an in vivo model. And as I said, the phenotyping across scales is very readily feasible. So we built, with help from David Rosenbaum and his group at Case, old colleagues, and the late David Rosenbaum, who was a fantastic collaborator and a prior colleague of Dr. Rudy's. We built a system to undertake optical mapping in the fish heart at a stage. And it's worth pointing out again, just for emphasis, that anything you want to do at scale, the real advantage of the fish is in those first 7 days when it can be done in 96 or 3D4 well plates. So we had to build something at a time when the whole fish heart is about 350 cells, if you include the atrium, and is about 100 microns in total length. So we built this system. We were able to very quickly do what we had said we wanted to do. We wanted to prove that we could actually identify genes that were relevant for repolarization. So we used optical mapping to do a screen of essentially mutants that might be resistant to dofetilide. We identified this panel of 50 mutants, all except one were actually feasible to pop into a network based on existing genomic and epistasis data from other platforms. And you can see here a cluster of genes of the membrane and the caveolus, including potassium channels, NOS1AP, which we identified prospectively about 2 years before it was pulled out of a human genetic study, as we did also with CCM3, which is another gene that was identified in the original studies, and not one, another important QT modulator identified in an unbiased way in human QT studies. Perhaps most importantly for us, we were able to show an interaction with the known genetics. So there were mutants available in KCNH2, the target of dofetilide. You can see here the heterozygote with this is the wild type in black, the heterozygote in gray, and then remarkable prolongation in the context of the homozygote. Importantly, you can actually see here also all of the electrophysiologic features. There's subtle prolongation with the head. There's triangulation and marked prolongation of the homozygote. There's even 2-to-1 AV block, which is seen in pediatric cases of QT. And then we were actually able, with voltage and calcium, to identify early after depolarizations at a point where, to be honest, there were no other models that really had definitive primary arrhythmias, and at a concentration of dofetilide that was about 10 nanomolar, so roughly equivalent to the circulating concentrations. And then, this is published, I'm not going to go into it in any great detail, we were also able to show, recapitulate almost all, in fact every drug-drug interaction that we tried, except for one which was due to the fact that it was not absorbed by the fish. But this is just one example, erythromycin and cisapride, with increasing doses of erythromycin shifting the curve for cisapride to lower and lower concentrations. We then thought that another strategy that we might try and do is see if we could actually use existing genomics in test cases to validate computational approaches to predicting gene function. This we did much later, but I think I'm putting it here because it's an important example of how you can actually use this scalable approach to rank every gene in the genome for its likelihood of causing a particular phenotype. So the traditional approach might be to manipulate each gene and explore the phenotypic universe. Obviously, the difficulty with that strategy is that, and it's been used both in mice and the zebrafish and a lot of others, is that here phenotyping is rate limiting, or the phenotype itself is not available in the model that we want to use. The alternative strategy that we thought we might use was to try and identify prioritized genes for investigation for a particular set of phenotypes. And here, the nice thing about it is you can actually do this in silico to start with the zebrafish. So we basically pulled out all the genes in the original null projects, and there were about almost 4,000 mutants identified. but with multiple different phenotypes. We did this for 338 phenotypes in parallel. We used the original phenotype data as a training set. We then used all of the existing genomic data, protein-protein interaction data, gene feature information, and a machine learning framework to try and predict which other genes in the genome would cause each of the 338 phenotypes. So, for example, in fact, one of the first ones we did was to look for things that affected calcium modulation and caused heart failure early in development. We were able to combine a couple of different strategies. This was work we did in collaboration with Fritz Roth, who was in systems biology at Harvard, and then subsequently went on to do some additional work with him looking at small molecule screens, which I'm not going to show you. But in essence, we combined a couple of different strategies, either a guilt by profiling, where it's simply things that go together, or a guilt by association, where there were definitive interaction data, either at a genetic or a protein level. And we actually used a combination score to rank every gene in the genome for these 338 phenotypes. We ultimately pared down the original number of training genes to only 767. And so you can see here that there was actually a deficit of training genes. And in fact, most of the data that we found that were valid here were for the two phenotypes that are most easily recognized, and that's cardiac failure and melanocyte migration, because those are obvious to inspection. And when we did that, we actually identified, and we were really heartened to show that we identified two out of the three genes that had been previously or subsequently identified as genes that cause heart failure in the general population. So HSPB7 was one. The second one was actually this TMEM88, which has recently been shown to be important. We weren't able to show BAG3 because it was actually in our original training set. That's the only other gene that's ever been shown at genome-wide level to be associated with heart failure. And in fact, you can see that we were able actually to do remarkably well. The bottom five are negative controls, and we essentially had all but one of our top 20 list were validated, although a couple of them, actually more than a couple, 5 of them ended up having to be done with really high resolution phenotyping before we were able to detect the abnormalities. But we note that the biggest problem here is, I think, the low resolution of the training phenotypes. So we're now going on to repeat this with genes that we know specifically affect cardiac electrophysiologic phenotypes, so that we can retrain the entire data set based on a repolarization phenotype that we're really interested in. And importantly, one of the things that we're also beginning to think about adding here are the addition of the ENCODE data, which help prioritize genes from GWAS or human data sets, and then, as I noted, more specific training sets. And then just to show you, this is HSPB7, which up until this time nobody actually even thought was really the real gene, but it does seem, and we have extensive data now showing that it actually has as profound an effect on calcium handling as does the IP3 knockout, which is immediately above it. And in fact, this seems to act as a chaperone for multiple proteins that are important in calcium handling. This is a small, it was originally labeled as a heat shock protein, but is actually a generic chaperone as well as having some heat shock activity. One of the things that we recognized fairly early that we wanted to study because it's accessible in the fish is the emergence of electrophysiologic phenotypes during development. So we've spent a lot of time trying to really understand what it is that happens. How does the heart go from this simple tube, which conducts, as you can see, incredibly slowly in the first instance, to, over the space of 48 hours, from a simple peristaltic pump to one that has really quite dramatic sequential action with, importantly, you'll see delay at the AV canal, something that you require in order to actually have a functional valve in evolution long before you ever actually see a valve. So you can see here, and then the whole ventricle will depolarize almost instantaneously, which is one of the things that allows you to actually generate the increase in pressure that is actually the mark of the closed circulation. So this delta is something that we really wanted to begin to explore, both for its role in arrhythmogenesis, but also for its underlying role in cardiac physiology. What we had imagined we would want to do, and what we've begun to do, and I'll show you some of the first steps in this, is to really build physiologic fate maps, to take voltage, calcium, mitochondrial potentials, hydrogen peroxide, redox species of other sorts, and essentially map them for every cell in the entire heart through the first 4 or 5 days of development, when we can still do it in 96 well plate format. to build maps for all of these things, including maps for cell-cell coupling. One of the things that we recognize very, very quickly is, and you can see here on the lower panel, that conduction remodels very, very rapidly. And in fact, I've mentioned to several people in the audience, although I haven't shown this because it's actually technically quite difficult to project, but there's actually this transition from the peristaltic pump with very, very narrow isochrones to this sequential contraction with, as you can see, the remodeling of not only AV canal but also ventricular conduction velocities occurs actually over the space of about 15 to 20 minutes, is associated, interestingly, and one of the reasons we've missed it for a long time, is associated with a transition in the emission spectrum of the voltage-sensitive dyes that we use. And so we believe fairly firmly that there's not only a protein transition at this stage, but a lipid bilayer transition that occurs, and it happens at about 43 hours. So literally between 43 and 43.5 hours, this transition will take place. We're exploring this in a lot of detail, but I think it's still not ready for prime time. I can show you some of the very preliminary data that we have offline if anybody's really interested. Importantly, this all occurs at the same time that there's a ton of remodeling going on. This is beta-catenin as a cell coupling and membrane marker. You can see here the cells not only change their coupling extent, but also dramatically change their shape during this period. In addition to the cell coupling, there's also a huge amount of remodeling goes on a cell-by-cell basis in action potential duration. You can see here the dramatic differences between the venous in blue, the arterial poles in red, that remodels again very, very rapidly, and this all occurs again in the space of only a few minutes during development. And you can see here by 48 and 72 hours, you have dramatic shortening of the ventricular action potential and some substantial shortening of the atrial action potential, as well as the emergence of all of the diastolic phenomena that we associate with the adult human heart. We would have predicted this really from the orthology between the repolarization biology in the fish at 48 hours and the human heart, which, as I mentioned, was something that really struck us as being remarkable because it's so much different than some of the rodent models, where the orthology is really quite different both at the cellular electrophysiology level and in terms of the drug responses. Interestingly, and I'm not going to go...
What is the typical heart rate?
Typical heart rate is about 110 to 150. The stage we're looking at, it ranges from about 24 hours, it's about 110 beats per minute, and by 48, it's about 130, 150. Its temperature is sensitive, but at 25 degrees, it's about 150 by 3 days.
And what about neural inputs and power?
No neural inputs until about day 7 to 12, depending on how you look at it.
And then there is a...
And then you can actually, I mentioned to some folks earlier, we've built a split GFP system with one half of the GFP in the neuronal, under a neuronal promoter, one half under a cardiac promoter, so that we can begin to really understand when those intercellular junctions form.
And you can really change the heart rate.
Yes, it can have a dramatic effect on the heart rate when they innovate. But it's interesting, a lot of the autonomic physiology is present, I think, because the dimensions, the distances are relatively short. So a lot of the neurotransmitters that you would associate in higher species with autonomic innervation are actually active in both directions, early in one direction, later in another direction, in early development in the fish. So we believe there's some real mileage in beginning to understand how that changes the heterogeneity, how it changes the baseline physiology of the heart, and something we're actively exploring. No, it's a really interesting topic, and one that is remarkably accessible. We can, as you might imagine, because it's transparent, we can use optogenetic tools to manipulate that one side or the other. We can ablate the neurons after they've innervated the heart. We can do really quite a lot of manipulations, although those manipulations I don't think we'll be able to do at quite the scale that we're talking about here, but they're definitely feasible. I'm not going to have time to deal with this in any detail, but suffice it to say that the fish is actually an important model in the adult setting for the study of regeneration. You can lop off the apex of the heart, and within about 60 days it will completely regenerate. And interestingly, all of the physiology that I've shown you occurs in the de-differentiated regenerate. So in order to actually populate the regenerative cardiomyocytes, we believe the entire heart has to actually de-differentiate electrically, partially uncouples. Cells migrate through into the apex, and then the whole heart recouples again, with the sequence being identical to that we've described in development. You can see here we began to think about ways that we could actually quantitate and estimate the heterogeneity of different cellular phenotypes. Much of this is feasible at cell or almost cellular resolution. And so we began to build coupling maps based on the direction and intensity of coupling, both for calcium and for voltage. Everything I'm going to show you going forward is voltage, but we've done the same for calcium. We're beginning to do the same for redox species and other parameters. And you can see here that the simple heart is really incredibly linearly coupled. There's unidirectional coupling, almost no lateral coupling. By 48 hours, you see this emergence of a pole of complexity in the greater curvature of the atrium and the greater curvature of the ventricle. And actually, people say, well, why is it at the apices of these curvatures? But actually, I think what it is that's why the heart curves. So I'm going to show you in a few minutes that the heart actually adds cells from both ends. And the reason that these curves occur in the direction that they occur is actually because of the asymmetry of mechanical and electrical coupling. And we have some data to begin to address this towards the end of the talk. You can see here there's a remarkable intensity and multi-directional coupling. We've plotted this. You can see how low is the amplitude and how unidirectional is the coupling at 24 hours. This is the ventricle here, summated in one. Almost all of these are frequency plots. These are conduction velocity and angle plots. You can see here fairly uniform coupling, particularly if you look at greater and lesser curvature. So VIC is the inner curvature, VOC is the outer curvature. And you can see here almost polar opposites in terms of the directions of coupling in these two territories. This is the inner curvature, this is the outer curvature. And this asymmetry, this polarity, we believe is actually, I'm going to show you some evidence of this, is a fundamental reflection of the different origins of these two groups of cells. But importantly, it is perturbed by a number of disease stimuli and a number of other developmental stimuli, which I'll show you. So remarkable diversity, and as I said, one of the interesting things about this is we can plot this in real time over the first 72 hours of development. In addition to this, one of the other things that we've noticed is that at a single cell level, you are very, very close to this. In addition to the coupling, you see this remarkable fragmentation. I'm going to show you a video of it in a minute, that is progressive. So initially the wave of depolarization is almost uniform across the whole heart, but by 72 hours it's incredibly fragmented, particularly in the apex. And the important thing to remember is at this stage there's only a single layer of cardiomyocytes. So this is not technically breakthrough from an underlying myocardial layer. We do wonder if it's actually breakthrough from an endocardial layer. We wonder if there is actually coupling at this stage between endocardial cells, and I'll show you the reason that we wonder about that. So this is just a demonstration of optical mapping in 72 hours. So this is with endocardium. You can see incredibly fragmented, and then it's back to this almost linear propagation pattern when we abolish endocardium. And it's possible to do this because we can actually build toxins. For example, this is one we've done with a nitroreductase that metabolizes metronidazole only in the cells in which it's expressed. So we add metronidazole to the well and we ablate all of the endothelium in the entire organism. And when we do this, you can see that you end up with, as I said, dramatic loss of this fragmented and highly heterogeneous depolarization wavefront, which almost, you can see multiple breakthrough sites. And yet this is still with a single layer. So we're now exploring, we actually built with Adam Cohen, who is a voltage sensitive transgenic recorder. We're now building an endothelial transgenic recorder for voltage and calcium in an effort to see if we can actually understand whether this is contributing to this phenotype or not. Endothelium is actually interestingly also required for maintenance, so we've gone on to do this. You can show that even though you can't completely knock down all of the endocardial signal, when you do this much later, you can actually, again, lose completely the wavefront fragmentation, the breakthrough phenomenon that you see. One of the things that you probably noticed early on, anybody who is used to looking at optical mapping data will have seen there's fairly obvious polarization of the ventricular phenotype. You can see here everything is pretty uniform from left to right, whereas by 72 hours there's this remarkable polarization which has no obvious electrical boundary at this stage. We looked to see if it might be related to differences in excitability, in coupling, or in cell shape, but we're unable to actually identify any of these. It's about a twofold to threefold difference between the inner and outer curvature, and it's associated obviously with coupling differences that I've shown you. And we believe that this is actually a phenotype that's important for generating the sequence of contraction in the ventricle. This slow conduction allows this part, the basal part of the ventricle, to actually remain partially contracted while the ventricle, the apex, is really undergoing very rapid contraction. And so it's much more sluggish, and you actually end up seeing this result in the coordinated contraction of the entire ventricle. We looked to see whether any of the traditional developmental polarizing pathways might be involved, and obviously one of the most important of these is the Wnt beta-catenin pathway, which we had already thought was important from work we've done in disease models, which I'll show you. The Wnt pathway obviously has a number of different avenues by which it can polarize cell coupling, not least through the effects on GSK3 beta and differential regulation of membrane versus cytoplasmic concentrations of beta-catenin and the intensity of coupling, but also through calcium and another pathway, the planar cell polarity pathway, which involves the segregation of discrete molecules that are specific to this pathway on different sides of the cell. So one of the nice things about the zebrafish that it lets you do is actually look at gene expression in the entire organism. So this is in situ hybridization. You can see here is Wnt11, and it's actually sitting in the pharynx, and then it moves into the heart. And actually Wnt signaling usually is of the range of only a few cell diameters. As you probably know, almost all Wnt signaling is thought to occur through a combination of a peptide and a lipid in a tiny vesicle known as an argosome that traffics in ways that nobody quite understands those few cell diameters, and then couples with a G-protein coupled receptor pathway that's based at caveoli, and then somehow results in downstream signaling through these pathways that I just showed you. You can see here, in addition to this, it's also one of the things that's quite interesting, also heavily expressed in the limb buds, which is a sure sign that it's important in cardiogenesis. Every gene that's expressed in the primary cardiac field is also seen in limb buds in every species in which it's looked at. In fact, it's one of the associations in human disease that's quite noteworthy. So we worked out which of the Wnt pathway members, there are existing mutants, and each of these was required. And importantly, the only Wnt that's in the heart that is required for this is Wnt11. Wnt5 shown here has no effect. Wnt11, interestingly, actually, what it does is it increases the coupling, and loss of Wnt11 increases the coupling in the inner curvature, decreases the coupling of conduction velocity in the outer curvature. So it seems to act as a tuning mechanism around this pivotal boundary, seen here in the mutant. We knew from work that we had done, and this is another illustration of the fact that you can actually study mutants that are lethal in any other species, because the fish survives by diffusion for the first 72 to 120 hours of development. So the L-type calcium channel, complete null, is actually viable until well after the heart is formed and well after cardiac activity has begun. You can see here, you actually see paradoxical increases in calcium transients in the L-type calcium loss of function. And importantly, these are abolished by things that inhibit SR calcium release. So you can see here, there still is some phasic abnormality or phasic transient of calcium in the L-type calcium channel when SR release is also inhibited, but most of this appears to be nuclear in origin and not cytoplasmic. But the interesting thing for us was we were then able to cross this lethal mutant into the Wnt11 background, and you can see here that paradoxically it actually rescues the Wnt11 phenotype. So loss of Wnt11 in the presence of a loss of function allele of the L-type calcium channel actually ends up with a dramatic effect, completely rescuing the Wnt11 loss of function allele. And that obviously suggested to us that somehow Wnt11 is responsible for downregulating the L-type calcium channel, at least at some point, in signaling in the ventricle. We'll look to see how this might occur. There's some modest effect on total immunoreactivity for connexin 43, but it quantitatively did not appear to explain the differences. Although when we then looked at the partitioning of connexin 43 between cytoplasm and membrane, you can see here there's a dramatic reduction in in membrane connexin 43, but you don't really change the total concentration of connexin 43 as seen here. The other thing to point out is that several other caveolar proteins are required for this. So this is NOS2A, which is the equivalent of NOS3 in the fish. And there are now we have 3 other caveolar proteins that also result in this dramatic effect on the localization of connexin 43. So the model that we have for Wnt signaling is that when Wnt signaling is on, the gap junctions are remodeled and tuned either by other endocrine, paracrine, or autocrine pathways or transcriptional signaling. But when Wnt signaling is off, then the entire system essentially reverts to a non-remodeled state. And importantly, what happens then appears to be dependent in some way on the L-type calcium channel. What I can tell you is that you can only rescue it with the C-terminus of the L-type calcium channel. But we're still not entirely certain that that's of biological relevance in this system, but there is a partial rescue with the C-terminus. What we then went on to try and do was to understand how the heterogeneity that we had identified in the early fish heart, which is you realize is a single chamber, how that might represent some of the heterogeneity that's seen in higher organisms. So for those of you who are not used to doing developmental biology, the heart forms from 2 primordia that are actually in an anterior crescent to start with the atrium more caudal, the ventricle more anterior. These migrate towards the midline to form a single tube, which becomes obviously the venous pole. This is actually mouse data, not fish data, but it actually, interestingly, is identical to what happens in all higher vertebrates. This is exactly how the human heart forms. And you can see that the ventricle here in red and the proximal part of the atria are from the original heart tube isolated here. But interestingly, in green posteriorly is this secondary heart tube, which actually becomes pharynx and then adds cells to both poles of the heart. So both at the venous and the arterial pole, you add cells to this heart tube. And we believe that this is what actually results when you have fixed elements in the greater curvature of the atrium of the ventricle and the looping of the heart. But irrespective of what it does for looping, the important thing is that these are embryologically distinct groups of cells. And they're added much later to the tube than all the rest of the cells in the heart. In higher vertebrates, where you have a 4-chamber heart, these cells form the majority of the right ventricle. A lot of the outflow tract in conjunction with some neural crest cells that migrate down from the arterial pole even later. But they're also involved in the formation of the SA node, some of the septum, and some of the elements of the AV canal. Interestingly, this had actually been thought to be unique to 4-chambered hearts, but we were able, as a result of the collaboration with Lenzon, to identify this marker. So this on the panel here is all cardiomyocytes. It's not a particular, you can see at this stage that the cardiomyocytes themselves do not express a huge amount of myosin. They're actually relatively modest in myosin expression, they're relatively poorly differentiated. But importantly here we have this marker Dracula, which actually excludes the proximal part of the atrium, excludes the distal part of the ventricle, and which we believed, at least we wanted to test it, was actually representative of the first heart flow. Importantly, you can see there's actually quite a lot of background from endocardium. This is a center slice, not at a myocardial level, but at an endocardial level. And you can see that it's also expressed in a restricted fashion to a set of endocardial cells that are also not the entire endocardium. So there's heterogeneity not just of myocardium, but also of endocardium at this stage. And this is the merge of the two. This is at the level of the muscle, and this is at the level of the endocardium. So you can see here in green the primary heart field with no green and largely blue at both ends and red at both ends. So what we began to look at were the genes that might be implicated in this, and suffice it to say, one of the nice things we were able to do is to use a mutant that doesn't have endocardium. So the fish have several mutants that have no endocardium. Cloche is the most important one because it's the most complete, and it's the most well known. And we were able to actually use both morpholemes and double crosses to show that in the situation where there's zero confinement from the endocardium, you can actually show, and we did quantitative cell counts to make it easier to see because it doesn't really project very well here, that with early labeling, with early loss of function in these alleles, that TBX5 actually has a substantial effect to reduce the total number of second heart field cells, whereas PIDX2, loss of function PIDX2AB, but not ABC and not C alone, actually has a substantial effect to increase the proportion of the heart that ends up being second heart field. And at this stage, you can count the individual cells, so it's actually quite useful. I'm not going to have time to go into this in any detail, but we did some genetic mapping. We had actually spent a lot of time in our human genetics working on the genetics of atrial fibrillation. We had actually, prior to doing any of this work, built a gene network for the genes at each of the loci that we thought were implicated in atrial fibrillation. You can see here some of the genes that we actually identified, PEDX2, LIT11, TBX5. And importantly, one of the most interesting things for us was that each of these genes, and we've tested all of them and several others that are known to affect second heart field cells in parallel, that they all affect not only the accretion of second heart field cells, but also the coupling of those cells. So that all of the mutants that eliminate second heart field or perturb first heart field, second heart field proportions disrupt this boundary, suggesting that this electrical boundary is actually an important surrogate for the final mechanical anatomical boundary in the zebrafish. So this electrical boundary in zebrafish presages the mechanical boundary in higher species. And in fact, one of the most interesting things is that you can actually see that all of these actually also have a dramatic effect on the directionality and the intensity of coupling, largely in this adventitial first heart field phenotype, the highly coupled greater curvature. So we imagine that somehow there's either a current sink or a developmental interaction between these two tissues that's required to pattern this boundary between the greater curvature and the lesser curvature of the heart, which is, we think, because it's regulated by exactly the same transcriptional cassette as the mechanical boundary is regulated, that we believe is regulated by these same genes in higher vertebrates. So having done all of this and continuing to work on the mechanics of this at a single cell level, we also wanted to go back to the original rationale for doing some of this, some chemical screening. In the last couple of minutes, I'm just going to show you what is possible if you think about disease modeling in this context. We obviously are constrained because of the fact we want to do this in 96 well plates if we're going to have any advantage at all. We're constrained to the first 5 days. So we took a condition called arrhythmogenic right ventricular cardiomyopathy. Again, one which many of you may know. Poorly penetrant in most instances, but in this homozygous form called Naxos disease due to deletions in plakoglobin, the C-terminus of plakoglobin, there are actually cutaneous manifestations which are almost 100% penetrant, even though the arrhythmia, sudden death are not quite so penetrant. There are also some contractile abnormalities late on in the phenotype. And importantly, in all of these heart failure traits, there's a really long lag between the onset of the phenotype and the eventual emergence of heart failure. We also were suspicious because of the genes that were involved that Wnt signaling might be perturbing this. So we began to build a model. We tested all of the known ARVC genes. At this stage, there were only 6, all of which are desmosomal proteins. We knocked all of them down in the fish. We showed that they were able to cause a myocardial phenotype with pericardial edema, cardiac enlargement, hypercontractility, an ultrastructural phenotype with loss of dense, electron-dense lines in the desmosomes, as you can see here. All of this is quantitated in this panel. We made a conditional germline mutant with using the Drosophila Gal4 UAS system, where we expressed, and we actually had shown that this was a dose-sensitive phenotype in the first place. These are the fish at about 4 to 5 months of age. You can see here's the wild type, looking fairly like a happy trout. And here's the mutant with the heart actually larger than most of the other parts of the organism. And you actually when you leave them even later, they develop peripheral edema just like humans. But at this stage, they're small, they're runty, and they actually have sarcopenia. They have a lower muscle bulk than the normal heart. This is reflected in the simple changes in the heart size, and these are also seen both in histology and in EM. And this is the very typical heart of the adult fish and the mutants, where you can see the whole thing is disorganized. You can actually see the white fibrotic tissue with the naked eye, and it's actually remarkably similar to what's seen in human hearts. Those physiologic abnormalities that we were able to detect, both in terms of heart rate, stroke volume, contractility. We went on in collaboration with a friend of all of yours, Andre Kleber, to look at the electrophysiology. We sized all of the cells. This is another thing that's feasible in the heart because there's a total number of cells that you can characterize. You can actually look at all of the structural and functional properties of the cells. We're able to show a substantial, in fact, almost 80% drop in the sodium channel, sodium current density at the membrane. This is it just classified in the wild type. In the control fish, overexpressing the driver line, but not expressing, overexpressing the mutant, but without the driver. And then when you drive the gene, you can see dramatic reduction in sodium current at the membrane. This is just actually a more detailed higher power diagram of it. In order to be able to screen with small molecules, we obviously couldn't do patch clamp or single cell recordings. We had to find a phenotype that was proximate, that was screenable at high throughput. So we actually looked at the traditional heart failure pathways. Again, you can see here's natriuretic peptide A and B, and you can actually image the individual chambers of the heart. Actually, one of the interesting things is that natriuretic peptide B is restricted to the greater curvature of the atrium and the ventricle. So again, another example of how there's uniquely heterogeneous cells within the heart, and this is actually already known from transcriptional work that was done 2 decades ago by Ken Block at Mass General. We actually did expression profiling, pulled out a whole host of genes, but it turned out that there were none of them that were better as a discriminant than natriuretic peptides. You can see here 15 to 20-fold changes in the expression level of natriuretic peptide. You can actually phenotype the fish heart failure, not in the ARVC model, but in many other models you can do it, even at the stage when it's in 96 well plates. But for this model, we had to build a luciferase reporter, which we could then move into 96 well plates. So here we have multiple fish with a disease, with a reporter in each well of a 96 well plate. We can then take small molecules in large libraries. We've screened libraries of up to 70,000 compounds for many of these phenotypes, pin transfer them into 96 well plates containing the embryos, and then essentially just look for fish that are alive and have normal natriuretic peptide concentrations. And so that's basically what we did. The colors have actually blended in this, I'm afraid, but in essence we looked for things that had where the fish were still viable by automated physiology, and where the luciferase done in the next reading was actually down in the normal range, so things that are in the 50 to 150 range. And when we did that, we identified actually 3 compounds, including this one, which is, actually I thought I'd like to label on, but it's SP217635. It's a compound that was originally developed as a GSK3 beta inhibitor by Smith-Kline-Beecham. When we add this, we discovered it a few days, so it's not surprising that it normalizes natriuretic peptide expression. That's how we discovered it. But importantly, it has a salutary effect on survival at 5 months. We've either left them untreated, where there's about a 30% survival at 5 months in this line, or we have treated them early, treated them late, treated them at intermediate time points, and you can see you can either prevent or treat the phenotype to reduce the risk of arrhythmia. Importantly, these are actually in the mouse model done in collaboration with Jeff Saffitz and Andre Kleber, where you can see here's the wild type in blue, here's the mutant in green, and here's the mutant plus the compound. So within several hours of adding this compound, you actually get rescue of conduction, of sodium current, and of calcium defects. There's actually a modest effect of contractility. It doesn't rescue contractility if you let the contractility emerge, but that's a very late phenotype. And it improves survival in 3 ARVC models. The original one it was identified in, and 2 others. And there's data from Jeff's group that it reduces the incidence of VT and telemetry in the placoglobin mouse model, although that's very preliminary data. This does not project very well at all, I'm afraid. In fact, I'm stunned at how badly it projects. I apologize. But suffice it to say, this is a control fish. We added SB2, this SB217635, and it actually leads to a modest increase in SAP97, SAP97, this target protein that's important for the sodium channel, and a variety of other proteins in the macromolecular complex associated with sodium. Even at baseline, there's an increase, but there's almost no SAP97 at the membrane in the mutant. This is the 2057 placoglobin deletion, but when you add SB2, there's rescue of this dramatically, and in none of these is there a total change. Is there a change in the total amount of SAP's 97 expression? So we believe that this is all centrally acting through trafficking on a variety of compounds, a variety of proteins in the complex. We're not sure which one it truly interacts with. We've tried, as I mentioned to some folks during the course of the day, we've tried a whole host of other GSK3 beta inhibitors, and none of them have this effect. So even although this drug is identified as a GSK3 beta inhibitor, we believe it's acting not just through effects on this domain, but potentially through much more complicated effects, which we're going to have to pull out the target or other targets in order to identify. It's worth pointing out that there's obviously unification between many of the proteins that we identified in our repolarization screen. So of the things that we found, LRP5 and 6, frizzled, NOS1AP, and now SAP97 as well as PSD95 are all involved in this caveolar signaling domain. So we believe that this is really a critical domain for the microenvironment in which the majority of the major channels seem to assemble. And so we're beginning to focus on systematic approaches to this in the zebrafish, knocking out each of the components, building genetic models using either TALEN or CRISPR technologies to actually be able to make true germline mutants for each of these and study them in epistatic fashion. One of the sorts of things we want to do in the future, we want to continue the phenotype screens that we've identified. As Dr. Rudy pointed out, cardiac innervation should be high on our list. Inflammation, for example, which is an important part of the proarrhythmic state, affects all of these pathways. Actually, WNT is an important modulator of the inflammatory state. What I didn't tell you is all of the mutants that we identified in our repolarization screen have a macrophage phagocytosis defect in null form in the zebrafish, something that wouldn't be picked up in any other system because they're all embryonic lethal. So the interesting thing is you need repolarization in order to survive. It's a really critical part of, as you might imagine, of restoring your membrane in multiple tissues. And as a result of that, I think there's some unique applications of many of these techniques to every tissue in the body. We're working with some new transgenic sensors, including one for voltage, cyclic AMP. We're trying to get a hold of a couple of membrane tension reporters that are available, FRET reporters that are available. And we're now also building subcellularly targeted calcium reporters for mitochondria, for nucleus, for Golgi. We're looking at new dyes in collaboration with Guy Salama and others. We're, as I said, incorporating a lot of the new genetic techniques, optogenetics for stricted dissection. I didn't mention any of this because it's more of developmental interest, but you can build mosaics for each of these. So we have hearts where we have 20% of the cells positive or negative for any of the factors that we've talked about. So we're beginning to look at how individual cells influence their neighbors during cardiogenesis. You can engineer almost anything you want with a variety of different things. I would love, having spoken to Dr. Nirvana a few seconds ago about this, to think about ways that we could begin to systematically approach non-coding. I think it's going to be incredibly difficult, but I don't think anybody has the phenotyping resolution that the electrophysiology community has to be able to truly explore this. I think it's something, as a group, as a community, we are really very well positioned to think about the entire phenome. I don't think anybody else can model computationally and integrate physiologically across all of the scales in the way that the people in this room can do. Obviously, new disease models for drug discovery. Hopefully get some of these to the clinic. But ultimately, our goal is to really think about a systems biological approach, as I've said, through iterative in vivo and in silico modeling with some of the people in the room. Obviously, it would be great to think about the ways to do this and to extend these findings to other tissues where the signal noise may be less clear cut than it is in the cardiac electrophysiology. I hope I've shown you that the fish offers a combination of in vivo cell biology with whole organism biology at scale, but it is worth remembering, as John Seidman, one of my old mentors, said, it is still just a fish. But I think we can learn a ton from the way in which the pieces are put together. I think we're beginning to explore the developmental basis of arrhythmogenesis, along with many others in the room. You can imagine the way that some of the other technologies that they're all working on could be implemented, how you could start off with looking at single channel, single domain moments in the membrane, go all the way up to human slices, and just in this room alone. Think about disease modeling in a much more systematic fashion, modeling all the disease animals in parallel, coming up with personal therapies. And really the potential, I think, is for a systems level approach, taking formal genetics of really complicated cellular phenomena that are only present in vertebrates, and trying to do that at every level, at multi-scale, doing the same with chemical screens, both for environmental interactions and also to identify chemical probes for many of these pathways, and ultimately testing this at systems level for arrhythmia and other things. This would not have been possible without several of the people on this slide. Andreas Verdick, who was actually a joint trainee between my lab and the late David Rosenbaum's lab. Gabe Bussa, who's a computational biologist in my group, and Eva Pluggy, who did a lot of the disease modeling. Randy's a chemist, Fritz is the systems biologist. Jeff and Andre and Jeff's group. David, I couldn't have done any of this without him. And then long-term mentors, John Pickett, Mark Fishman, and Bill McKenna. And I have to say, I've gotten a huge amount of mentoring and input from everybody in the room that I met with today, and thank you very much for hosting me. I should also mention my funders. And this is the dark and cloudy day that is beginning to emerge from the window looking over Boston in the last couple of days. Thank you for your attention.
Questions and comments?
That was really nice. Did you try lithium in your area?
We didn't. Yeah, it doesn't. It has the predicted effect. So that was one of the GSK inhibitors that we tried. And even although lithium can be titrated much more precisely than any of the others, we cannot rescue. We've tried actually we spent a lot of time with lithium. Because SB217635 was originally identified as a therapy potentially for black hole elements. We've not been able to replicate.
So do you think it's wind then, or do you think?
I don't think so. I don't know what it is. I mean, we tried this in a relatively unbiased fashion, so we're trying not to bias ourselves. We know there is a Wnt effect. There is at least some of the effect is mediated through GSK3 beta. We can look at other mutants and show that they have some effect on the phenotype, but they don't elicit the electrophysiologic effect, and they certainly don't rescue the whole phenotype. So we're beginning to do a SciLac to try and identify the other targets of this compound to see if we can work out exactly what it's hitting.
And does it rescue the fibrosis in addition?
Completely. It doesn't. It depends how far along. If you're very far along, you can prevent the fibrosis all the way up to about 2 months. After you get a lot of fibrosis, it's irreversible, and that's when the contractile failure comes in. So if we give it early, we just prevent everything. The fish. In fact, we can actually show that if you give it for the first 10 days of life, you can rescue the fish for at least 6 months. We do see a phenotype emerging much later on, but there's a window when all of this is going on, which for us is actually quite interesting. It coincides with adolescence and early adulthood in humans. So, which is interesting because many of the arrhythmias that were in this syndrome actually occur at that stage. We don't honestly know what it is, but we think, again, even if we don't have a drug, we've got a chemical probe that will let us work out what the other elements of the pathway really are. So it's an exciting thing for us to work on for sure. Sorry, you had a question.
Yeah. If you look at the human ventricular wall, then it's mainly composed of compact myocardium and a very thin layer of trabeculi.
Yes.
And if you look at the adult zebrafish heart, it's mainly trabeculized with a very thin
layer of compact. You're right.
So how would that affect the mechanism of arrhythmias, and how representative is the ventricular myocardium?
Yeah, that's a very good question. So I think the answer is we don't know. We know that. So for example, we recently cloned a couple of human genes that cause left ventricular non-compaction in humans, and interestingly, they reproduce the same phenotypes in the fish. You can see exaggeration of compact myocardium. So there is of trabecular myocardium and loss of even the tiny amount of compact myocardium that exists. But I think you're right. I'd be wary of making too much of an orthology beyond the level of integrated cells, but it's something we just haven't explored. We do recognize, I mean, for example, there are a couple of things. So a lot of the acquired phenomena in intercellular junctions, so compaction, mechanical compaction of the heart, actually looks like it's a function of mechanical load earlier in development. So we're actually beginning to study the role of increased load in the zebrafish heart to see if we can increase the compaction in the fish heart to model the mouse of the human heart. And in fact, when you look at the embryonic mouse, many of the phenomena that we see in adult human actually occur in the first 7 days of mouse life. So we think there's a combination of genetics, epigenetic factors, and I would be the first to say I don't imagine that it's a perfect replicate of adult human, but I think we'll learn a lot from understanding how they differ and where the nuances are in terms of the discrete types of myocardium and how important they really are. How important is the packing of the myocardium as opposed to the individual cell-cell junctions or the heterogeneity.
A lot of arrhythmias occur in the compact wall.
Pardon?
A lot of arrhythmias occur in the compact wall. Which
I agree.
Absolutely.
And those same arrhythmias, I think, occur in the compact wall of the fish, at least the ones we looked at. But I'd be the first to tell you, I don't want to make a comprehensive claim that it reproduces everything, not for sure. And in fact, to be honest, most of what we've done is very early on. Trabeculation doesn't really occur probably for about another 3 days after most of this was done. But the interesting thing is actually the trabecular myocardium looks like it's based on this epithelial mechanism in compact myocardium. So a lot of the epithelial signaling that you see early on in the fish may actually be particularly important for compact myocardium. That's one of the reasons we believe that we may have a better model than we imagined, just simply, even at the early stages, for compact myocardium dependent arrhythmias. But we need to explore that much more systematically. I completely agree. And look at a variety of different arrhythmogenic substrates and see if they're really present in the fish in this way. And in fact, one of the interesting things I'm looking forward to doing is taking some of the insights that folks in the room have gotten from ECGI in human arrhythmias and then in human slices, and then bringing those back into the fish to try and validate exactly the points that you bring out. I think it's an important point. If we're going to use this for drug screens, we need to really understand the mapping of not just the genotype, but the phenotype back onto the fish. Completely agree. Great question.
Have you thought about, in the experiment where you ablated the endocardium, is it possible that the myocardium is de-differentiating? For example, if you lose neuregulin or those kind of factors, and does it phenocopy that?
Oh, 100%. We can actually, I didn't show it because, actually just because I didn't put the slide in, but I can show it to you. So neuregulin rescues this phenotype perfectly. But the interesting thing, so we've begun to look at this. There's actually, I don't know if you're aware of this in the neuroscience literature, there's an amazing effect on micro-ruffling of membranes by neuregulin in synapses. And we've looked at this before with EM and not seen anything, but we're now going back to really focus in a very concerted way whether that's what's happening here, that actually it's just changing the surface area of the membrane interactions, and that actually is dependent on neuregulin. The other thing that it's definitely dependent on, which I didn't show you, is flow. So there's an element of the endocardial phenotype is dependent on the stimulus of flow, then transmitting some signal to myocardium. And in fact, when you look at almost every other tissue, brain, pancreas, liver, this is a common theme that almost all of the radial coupling of every cell type in every tissue out from beyond the circulation is dependent on flow. So there's some really nuanced way in which that polarizes tissues, and that's essentially what cell coupling is. It's how do you organize a tissue in a way where the functions are orchestrated in the right direction, and that's, I think, what flow does is it sets one of the extremes in that equation. So it's an interesting phenomenon that we're trying to explore in other tissues.
And could you please elaborate on the requirement at the organ level to de-differentiate during the adult regeneration?
Yeah.
What did you say?
So that's certainly the heart. I don't know about other organs. So we did this work in conjunction with Cam Poss And we looked at a variety of different markers. You get upregulation of some of the major transcription factors. You get partial uncoupling of the whole heart. You get, I mean, it's quite dramatic. You should see, in fact, if you want to see arrhythmias, you should see what it's like when you lop the end of the apex of the heart. They really get this sort of amazing, slow VT. And then it all settles down and they recover and they recouple. And the heart basically is back to normal. So we've shown very convincingly that this occurs in multiple different injury models, that no matter how you injure the fish heart, without scar, if you have scar, you end up with a scar. And that's something we're beginning to look at. But if you don't have scar, the whole heart will de-differentiate, and that's dependent on a epicardial and pericardial signal. And then cells migrate, cardiomyocytes de-differentiate, they duplicate, they migrate into the coagulum at the apex of the heart, and then they themselves differentiate into cardiomyocytes, couple, and then the rest of the heart recovers, and then it seems like you need to have the new regenerate and the base of the heart sort of coupled together. You can't have one couple without the other. So we've been trying to see what happens if we manipulate the coupling in the existing part of the heart and whether or not that interferes with the ability of the new regenerate to integrate with the rest of the heart. And those experiments are not complete yet, but we're working hard to try and do that by switching on and off so that the dominant negative connects and you can induce or switch off in this system. And so we're trying to see if we interfere with coupling in the differentiated part of the heart, does that prevent the regenerate from coupling to the rest of the myocardium?
And do you know that the cells that regenerate, the de-differentiated myocytes are in fact the myocytes that are de-differentiated and are not
So you can do
resident stem cells?
Absolutely. So we've done, well, actually, this was work that Ken Post did. We've subsequently done it in our models, but we've done lineage fate mapping, and you can show that all the cells in the regenerate came from cells that were originally, before the injury, were originally differentiated cardiomyocytes. So all the myocytes at least come from that. That's pretty, I mean, could there be a small population that's from resident stem cells? I'm sure there could. We certainly wouldn't want to say we'd exclude it.
Any other questions or comments? The hour is getting late, so thank you so much.
Thank you very much.